Okay, now hopefully I'll get this thing to work the way I want it to today. Success. Third time lucky. We're talking enzymes and other molecules, particularly enzymes with relation to the sac that's coming up. And as we discussed the other day, we're looking at metabolism, anabolism, catabolism, enzymes, and a whole bunch of organelles which are involved in enzymes being made, that protein synthesis, which we'll discuss in more detail in a later chapter. So, interestingly, that you didn't actually understand this term the other day. So metabolism, all of the chemical reactions that take place in your body. Now, we talk about 30,000 genes coding for, therefore, 30,000 proteins, many of which will catalyze reactions. We've got a lot of chemical reactions going on all the time. So, some of them we can describe as anabolic, and therefore they build substances. And the classic example of that is photosynthesis. And you'll enjoy my lisp as the year goes on. And then there are catabolic reactions. And the classic example again, of course, is cellular respiration. And those two chemical reactions are critically important throughout the year because without both of them, which occur in prokaryotic cells, living in endosymbiosis with our eukaryotic cells, without those two processes, life on this planet would simply not exist in the form we know it, if at all. Photosynthesis has allowed us to, in fact, gain oxygen in our atmosphere, and cellular respiration allows us to take the product of photosynthesis and release the stored energy within it. Otherwise, it's of no value. We discussed in class how one major misconception students have is that plants have chloroplasts only and animals have mitochondria only. Now, animals only have mitochondria, but if you have a chloroplast, you've got it there for a reason. You're using it to capture light energy, create chemical stored energy, and then release that via cellular respiration. So you must have both. So plants have both of these organelles. Clearly, all these reactions require many hundreds of enzymes. And often, some of these more complex reactions require many enzymes working together to produce the outcome. And so we'll talk about much more about those two particular reactions later in the year because they are very, very important. But in the build up to our sac, of course, these are the important things we need to include in our background. Enzymes are biological catalysts. So they're substances which, so catalysts are substances which cause a chemical reaction to occur faster. In the case of our living creatures, enzymes are biological. They belong to living things. But we know quite a few things about them. They belong to that larger class of biomolecules called proteins. They are specific to a substrate, or more correctly, to a group of substrates of very like chemicals. And we'll talk more about that in a moment. They can be reused. So as long as there's substrate present and the active site isn't damaged, an enzyme will keep on working. So therefore you only need them in very small amounts. You don't need to produce lots, which is good. Most activities of a cell are about conserving energy at all times. So making lots of enzyme, really expensive process. You don't want to do if you don't have to. They are neither reactants nor products. So they're not part of the chemical reaction in a meaningful way in that sense, but they are used to speed up the central part of reaction, that actual movement from reactant to product. And I drew that for you on the board, and unfortunately I can't draw it for you here. I will learn how to do that one day. They reduce the activation energy. So the activation energy is the amount of energy required for a chemical reaction to start. A catalyst will make that amount of energy much less, well, the requirement much less, and therefore they reduce that activation energy. They can catalyze in both directions, so they can work anabolically and catabolically, depending on the conditions. They don't change the direction of the reaction though. They respond to the other chemical signals to decide which direction they're going in. I have to pause for a minute, sorry. Okay, I'm back and I forgot what I was up to. What was I talking about? Let's see, we've gone back to the start of the, oh, here we go. Oh yeah. So they don't change the direction of a reaction. They actually respond to the chemical situation and move with the direction of the reaction. They don't change the final amount of a product. So if there's 100 grams of substrate, then we're going to get however many grams of product that gives us. They can't change that. They make it happen faster. And they are affected by certain conditions, which we'll go into in a minute. And there's a couple of diagrams at the bottom showing both the lock and key and induced fit models, which we'll talk about in a slide soon. So this is what our SAC's about, of course. There's two well known and well studied and therefore well examined characteristics which can change the ability of an enzyme to work. One is temperature, the other is pH. Your SAC's about testing one of those. So all enzymes have a preferred temperature range. Below the optimum range, they tend to become dormant and work slowly. And so you can see that as we did the other day, as we warm the solutions up, the reaction becomes more obvious and they're much quicker. If we go beyond the optimum temperature, we actually cause the chemical to change shape. It physically reacts to that heat and changes the shape of the enzyme, of the active site. So that the active site is where the enzyme connects to the substrate. If the active site changes, it can no longer connect to its substrate, and we call that a denatured enzyme. It's finished. The cell will try and recover amino acids from it and recycle materials, but it's never going to do the job it was meant to do until it's been remade. A similar thing happens with pH. However, with pH, the optimum range might be, in this case, looking at a graph which would probably suggest a neutral pH, and so between 5 and 8, it probably works. Below 5, becoming more and more acid, it denatures. So there's a chemical reaction with the active site which changes its shape. Above 8 pH, it also denatures. So there's no dormancy here. There's a range within which it works, and outside those ranges, there's no activity because the active site has been chemically changed. It can no longer bind to its substrate. Okay. We also talk about the idea of affinity. So we want to, many organisms want to be able to control the speed at which catalysts work, which is strange considering we're using catalysts to increase the speed. But sometimes, as we'll see in the experiment next Thursday, the reaction can happen quite quickly. I think Mr. Boson was saying that catalase produces, 40,000 or was it 40 million reactions per minute or something, which is enormous, it's huge. So sometimes you want to slow that down because the byproduct you're producing is toxic in itself and you won't be able to deal with the byproduct, so you've got to slow stuff down. So certain chemicals can be used to inhibit the action of an enzyme, and so they can either block and compete at the active site, or, yeah, that's probably how they work basically. Coenzymes and cofactors can also assist by speeding up the work of an enzyme or competing with it again. And simply the amount of reactant present will have an impact on how quickly an enzyme will break down that substrate. So we have these two models. Excuse me. Lock and key model is a classic model. It's been around for a long time. In the sense we see an enzyme having an active site, the lock, that fits the substrate. So the substrate becomes the key into the lock on the enzyme, and therefore an enzyme is substrate specific. Is used on one substrate only. However, more recent research is suggesting that model is not adaptable enough, and that a lot of research is now showing that enzymes have the ability to in fact change to match an active site. Now, we're not talking about an enzyme therefore becoming capable of doing anything, but rather, I think I used the example in class the other day, we might have an enzyme specific to disaccharides. So the active site on the enzyme can be combined with a range of other disaccharides, not just one, or it might be able to break down a range of complex carbohydrates. So it's one class of chemicals that it can act upon, because the active site still needs to have enough affinity with the substrates binding with, but it can adapt to fit it, which probably makes a lot of sense, really. So how do we make an enzyme? How do we make a protein? of course. Now, we haven't done protein synthesis in any detail yet, and we will. I think it's chapter 4. Yep, I think it's chapter 4. So, in the nucleus, we have, of course, in humans, 46 chromosomes containing all of our DNA. On those 46 chromosomes, we have roughly 30,000 genes, which is a lot less than we actually thought originally. Each of those genes has the information for one protein to be produced. I saw a little, what do you call it, cartoon strip showing this the other day with the U9 thing I was doing, and they had the library, and in the library there was all these books, and on a page of a book you had the gene. So you photocopied that information, which is the role of the messenger RNA. You take that information out to the ribosome, which they put up as the kitchen, and you cook the protein. So it wasn't a bad analogy, but essentially the nucleus contains information. The DNA can't leave the nucleus, so we copy the important parts onto messenger RNA. Messenger RNA goes to the ribosome. Transfer RNA comes to the ribosome with amino acids attached, and we have this complementary base pairing between messenger RNA and transfer RNA, and the amino acids join together into a long chain of polypeptide, a long chain of amino acids called a polypeptide chain. That polypeptide chain is essentially the first level of the protein. It moves into the endoplasmic reticulum, where it's folded. We get the alpha coils, we get the beta pleated sheets, and of course, most importantly, we end up with an active site. If that's a smooth endoplasmic reticulum, then it will now be transferred into the cytosol. It may do some work on the membranes or do something within the cell. If it's a rough endoplasmic reticulum, the belief is that it then goes to the Golgi apparatus for either further synthesis or packaging. If it's packaged for export, that then is put into a vesicle. The vesicle travels to the cell membrane. The cell membrane absorbs. The vesicle sort of becomes part of the cell membrane, and that protein is released outside. Now we'll do that in a lot more detail later this term. But that's protein synthesis in a nutshell, and you probably need it for your sac, so it's worth sort of maybe looking ahead and thinking about it. Lysosomes in animal cells and vacuoles in plant cells are able to store a lot of this material. So lysosomes are involved, of course, in apoptosis, as we've already discussed, and we'll discuss more of, but they can also store things for later use and release stuff that isn't going to destroy the cell. And vacuoles in plants do the same thing. Not only are they full of water and applying turgor to the cell wall and the cell membrane and creating a nice firm cell, but they also store a lot of chemicals that the plant cell may need later, including the outcomes of protein synthesis. And so I'm going to step past these. I've actually just described all that without necessarily going through it, and I realise this video is going to run out if I'm not careful. I've just done it. I've succeeded in getting in before 15 minutes. Well done me. Even the phone call in the middle. Okay, I hope that helps with chapter 2, and particularly with your sac, and I'll see you guys next week. I've almost got it. No, stop.